Uh, the frustrating thing for me, at least over the past 20 plus years, has been that as much as we acquire a lot of data with magnetic resonance imaging, the phenotypes that we can derive are, of course, not sufficient to understand what's going on at a cellular level. And to bridge that gap, we have we and other groups have come up with using transcriptomic data to bridge the gap. As a way of background, as you all know, cerebral cortex at a cellular level is comprised of many different cell types, of course, neurons, pyramidal cells, and interneurons, and many different non-neuronal cells, in particular astrocytes, oligodendrocytes, microglia, but also other cells such as endothelial and neural cells. Now, of course, we cannot see those cells on our MRI scan, so to bridge that gap, as I said, we are using transcriptomics data. And we are fortunate to have data on gene expression in the human cerebral cortex from a few donors, but I don't have time to get into the reliability of the profiles of gene expression that are available for each of the 34 regions that we segment with FreeSurfer. And then the trick is to correlate those interregional profiles, for example, interregional profiles derived from MRI about cortical thickness or surface area with interregional profiles in gene expression. And that's an example of such a correlation. And then, of course, evaluating statistical significance of that correlation across multiple gene expression profiles. Now, of course, we are not working with any genes. We are working specifically with gene markers that are cell-specific, specific to different cell types listed here. Here is an example of 9 key cell types. And the gene markers come from single-cell transcriptomics that was carried out by Cerebral in this case almost 10 years ago in the mouse cerebral cortex. So again, we are correlating interregional profiles of gene expression in the cerebral cortex of these cell-specific gene markers with interregional profile, in this case, of thickness or surface area. Let's start with thickness, and that's a result of our collaboration with Enigma, where we were interested in group differences in cortical thickness in 6 different disorders that are listed at the top. And through that collaboration, using data from 140 plus cohorts, we were able to calculate first those profiles of group differences and then run our virtual histology on those cortical profiles. So let's first have a look at how those profiles behave. Here you have, on the right side, you have the number of cases and controls for the different disorders. For example, here at the bottom, group differences in cortical thickness for patients with schizophrenia as compared with controls. And you can see that, as expected, cortical thickness is lower in patients. That's why the values are below 0. But you can also see that the difference is not the same. The line is not flat. So in some regions, the differences are larger than in other regions. And you can see even here that there is a certain commonality in that the regions with large differences in schizophrenia also show somewhat larger differences in other disorders as compared, let's say, regions in the occipital cortex. So here we have the very first commonality of interregional profiles across different disorders. We can compare this phenotypic commonality or similarity across disorders more formally and compare it with genetic similarity that has been known for quite a while now based on large GWAS studies. When we do that, so here you have the correlation matrix of the phenotypic similarity in the group differences in cortical thickness. Here the matrix of genetic similarity. When you plot the 2 types of similarity against each other, you see that many of the disorder pairs line up fairly well along the regression line. Some are outside. So, for example, much less similarity in the genetic and phenotypic variations between ASD and schizophrenia, as an example. But overall, one type of similarity explains about 27% of the other similarity. Now let's move to virtual histology. So here we see that, in fact, across all disorders, the group differences in the interregional profiles are correlating with gene expression profile or profile of the expression of genes specific to a certain type of pyramidal cells, and to a great extent also to astrocytes and microglia, in the direction that regions with high expression of those cell-specific genes are showing larger differences in cortical thickness. Now, again, we see a similarity here in this neurobiology. We can evaluate it more directly by running principal component analysis on the group differences and relating it again, and we see that PC1 is again strongly related to the neurobiology, to the gene expression related to CA1 pyramidal cells. And we can go beyond that and identify other genes that co-express with the genes that are related to our commonality, the common profile. For that, we use a different data set here of gene expression across about 500 donors. And when we plot those genes, we see that there are essentially two types. One set of those, a total of 400 genes, one set is highly expressed before birth and then the expression goes down. The other one shows the opposite profile, low expression before birth and relatively high expression throughout the adult life, at least the span that is sampled in this particular data set, up to 40 years of life. And the last question we ask, what are those different genes in those two groups? For this, we use gene ontology. And not surprisingly, of course, the ones that are expressed before birth are mostly genes that have to do with the development of nervous system, including, for example, dendrite development. The postnatal genes are more related to actually functioning of the cerebral cortex, or in specifically neurotransmission, but also regulation of neuroplasticity. So here we have one phenotype, cortical thickness, group differences in cortical thickness, and the underlying neurobiology pointing in this particular case, and we have some other data that support this conclusion, pointing to the importance of dendritic branching, both from the developmental point of view and from the plasticity point of view. Now, let me move to the other phenotype that is very different. Now we'll be looking at cortical surface area. Why do I say it's very different? Because in fact, as you can see on this slide, most of the tangential growth, which is indexed by surface area, takes place either before birth, both before birth and during the first 2 years of life. So if I look at surface area in my brain, it's probably very, very similar to what it looked like by roughly the age of 2 to 4. So it really provides us with a nice developmental window into very early brain development, and that's different from the point of view of thickness. Thickness, as you know, is changing dramatically during childhood and adolescence, but then continues to change somewhat less in late adulthood. Now, if we want to use again gene expression trick to get at the neurobiology, need to use a different type of genes, and for that we use cell-specific marker genes for which we have values of gene expression from roughly mid-gestation, fetal cortex. And that's available only in 11 cortical regions rather than, as I showed with virtual histology, in 34 regions. And the data come from two sources: the single cell RNA data that will tell us what are the good gene markers for the different cells come from this work by Baduri and others using fetal tissue from about 6 to 22 post-conception weeks. And see here, for example, and we'll be paying attention to radial glia and to intermediate progenitor cells. But of course, we are examining all the other cell types as well that are present and can be therefore assessed in mid-gestation. And then we will be relating profiles of gene expression of those cell-specific gene markers obtained in bulk RNA by Li et al., this publication in Science 2018, again from about the same period of mid-gestation, 10 to 22 post-conception weeks, and we'll be relating it to surface area of the same 11 cortical regions. And we again carried out this work in the context of Enigma Consortium, but we also used the ABCD study, but we binarized it to individuals with the top 20% score on psychopathology, child behavior checklist, and bottom 20%. So we have again the case control design here. And we then brought together, again, 100 plus cohorts with these data and ran virtual ontogeny on that profile. So first, let's have a look at the profiles. Here they are shown for those 11 regions. The regions on the left, we can globally call them multi-modal regions. The regions on the right are mostly unimodal regions. Now, if you are above 0, it means that controls have higher surface area than cases. So you can see very clearly that that's the case for ADHD, for schizophrenia, also for that split ABCD study, and much less so for the other 3 disorders. You also see that there is a profile. The group differences seem to be much larger on the left for these regions and less so for the regions on the right. We evaluated more formally with cluster analysis. You can see that those multimodal regions cluster together, the unimodal, with some exceptions, also cluster together. And this shows again that the multimodal regions show bigger difference than the unimodal regions in the case of ADHD, ABCD study, and schizophrenia. Now, when we do our virtual ontogeny, we see that it's mostly the undifferentiated progenitor cells where the cell-specific markers of those cells, their gene expression, correlates with the profile of group differences in surface area, such that regions that have high expression of those genes are showing larger group differences. We also looked at an intersection between these genes and genes that are involved in a number of risk factors, perinatal risk factors of psychosis. And you can see here that when we look at that intersection, genes that are related to developmental growth, birth weight, and congenital malformation, there the intersection is quite strong for radial glia. On the other hand, when we look at genes that are related to maternal hypertension and preterm birth, those genes intersect mostly with the vasculature genes. So we see 2 different pathways that may go towards lower surface area in relation to the early brain development. And very quickly, the last part is really about other genes that may be shaping cortical growth, the genes that are not typically addressed in our JIVA studies, namely copy number variants, and we'll be focusing here on deletions and duplications. The advantage of this approach is that there is a clear relationship between gene dosage and therefore deletions, duplication, and the expression of the product here. So you can see that relationship. And with my collaborator and my postdoc, Jitye Liao, we have done this in 3 cohorts in Sargon EU Study, Imagen, and UK Biobank. We have done it using so-called burden score, when we add up essentially copy number variations across the entire genome, separately for deletions and duplication. Here I'm just showing that those are distributed across the whole genome, of course, in relation to the size of the different chromosomes. And for any given individual, we may see up to roughly 10 deletions across the genome. So in the majority, of course, we see none. And now we'll be relating this burden, the total number of deletions or duplications, with surface area across the 11 regions. And you can see that that burden of CNVs is very clearly predicting lower surface area in case of deletions, much less so for duplications. And when we do virtual histology, we again see very strongly this relationship with progenitor cells, with IPCs, but also excitatory and inhibitory, but going in the opposite direction. The different undifferentiated and differentiated cells are showing different directionality of the relationship, in that again, for the progenitor cells, regions with more expression specific to these cells in mid-gestations are the regions that are showing larger effect of CNVs. And the final point, we were interested in looking at those genes, trying to get a better understanding of what they are. We classified all genes we pulled out using gene expression, genes that are showing high co-expression with our progenitor cells or low expression with the progenitor cells, and then reran this analysis. And it's very clear that if the genes are very close to the progenitor cells, the effect is very large, again, mostly for the multi-modal regions, not so for the unimodal regions. And the opposite is true for the genes that are far away, are less co-expressed with progenitor cells, with the exception of the primary visual cortex. And that's a very interesting region that very often shows up as showing very different architecture. So to finish up, it seems that we definitely see a lot of common neurobiology that underlies structural properties of the cerebral cortex in psychiatric disorders, in case of surface area, perhaps related to the pattern of the co-expression of progenitor cells versus differentiated cells. And I'll stop here by acknowledging the contributions of my postdocs, Yash Patel and Jitye Liao, and my collaborators at SickKids. Thank you very much for your attention.